Okay, let's just 1 minute. Let's just go ahead to start. I don't know whether I mentioned that before. Next week, Thursday, November the 6th, I won't be here, but Dr. Scott Baldridge will give a guest lecture about parasite. And I offer 5 points, extra points for the attendance. So please come in, okay? Okay, let's talk about something we did on the slides 2, which is lecture 2, but we will be go to the next chapter. So we just go over what we talked already, just do very briefly. Okay, we have a glucose. We going through glycolysis, generate pyruvate. And this process we call it glycolysis. And how many ATP generate? It should be 2 ATPs generate as an individual cycle. But if this is part of the aerobic respiration, I'll just say individual, that will be 2 ATPs. 8 ATPs will be part of the aerobic respiration cycle. Okay, then the pyruvate right here will go in a transition step, generate acetyl-CoA, and here happens is NAD reduced to NADH2. And then the acetyl-CoA going to go in through TCA cycle. And then you should remember there is a couple places happened is NAD reduced to NADH2. And then also have a place happens is FAD reduced to FADH2. Also here will be NAD reduced to NADH2. And don't forget, there is a GDP becomes GTP. So how many ATP created? There's 8. This is 3, 3, 3, 1, 2, and 3. So 3 ATPs. Multiple by 4, add 2, add 1, this is equals 15 ATPs. And we have 2 cycles, because this is a branch, so we have 2 cycles. So this is, add up together is 30 ATPs through TCA cycle, including the transition step. So you add up these together, a complete aerobic respiration, there is a 38 ATPs. And I said this is the key, the core knowledge of this session. Then we wanted to talk a little bit more, which is how the ATP going to be created. This is called substrate level phosphorylation. But majority of the ATP created here related to NAD and FAD, we call oxidative phosphorylation. And remember, this is related to when we talk about transfer phosphate potential or potential phosphate transfer power, and here related to chemosmosis, which is Peter Mitchell in 1932 mentioned about the theory and win the Nobel Prize later on. Okay, so very brief, we talk about what we discussed last section. Now I also mentioned if you want extra points, then every single chemicals, the name and the structure, you should be well defined. And one name will be 1 point, and the structure will be 1.5 points in the exam. So that will be plenty of the lots of the huge amount of the extra points for you to make up if you did not do very well in the exam 1 or exam 2, okay? So that's what we do, a very brief review, what we talked. We left one slide we haven't talked. So there is a side reaction, we call it pentose phosphate. The procedure is right here. We don't want to go over it in much more detail. Pentose phosphate, it will generate a couple of important precursor material. One is a reducing power is the NADPH. That is finally can be used in the Calvin cycle, which is related to anaerobic, anabolism instead of catabolism. And also more important, they will generate a 5-carbon sugar, which is ribose. And the 5-carbon sugar ribose is a major component of genetic base, DNA and RNA. So it is important, but we will not talk very much detail because we want to move on to focus on the next big chunk of the material, which is we talk about the genetics of the bacteria. Okay, so this section we reviewed here. We're going to move on to the next section. Today we're going to talk about the DNA and replication and the basic knowledge. Then we're going to move on to RNA transcription translation later on. So microbial genetics. The outline of these two classes will be talk about is DNA going through replication and then transfer the genetic information, go to RNA. This is called transcription. And finally, it will become information of protein, started with amino acids, then composed by peptides, and have a different structure, finally become a protein, and this is we call translation. And this blue line is very important in the genetics area, so we call it central dogma. This also will be the outline of this lecture. Now, if you learn already, this could go back. RNA could go back and become DNA. That's called the reverse transcription. So, and related to reverse transcription, in the old time, we have the PCR, we call it a reverse transcription PCR. Because for some of the material, it's difficult to get the DNA or get purified DNA. We can get the RNA, then going through reverse transcription, we get the DNA. Okay, so this is blue line, and it is outline what we're going to talk. So let's talk about the DNA first. Before talk about that, we want to go over the history. It is very important. We know the history. The development in the 1920s to 1960s or 1950s in the history of the molecular microbiology and the biology and the microbiology, people start to working on some very magic stuff. Because of the time limitation, some of the stuff I will be briefly, because used to be, this is a separate lecture, because I don't have that much time with our fall break and also a couple classes are canceled, so we're going to be a little bit brief. Okay. So in the scientific area, the scientists started to looking for genetic information is as early as 1920s. The reason is, in 1912, there is a big outbreak or pandemic called the Spanish flu. The Spanish flu kills millions of people in the European country. So people want to find, first of all, is a pathogen, which pathogen caused Spanish flu, and also want to do something more to find the genetic information. And by the time in the scientific area, They are trying to find the transformation material. So they call transformation principle, which means which chemical is important for the genetic transformation. And then they do lots of the research. So there is a couple research are very important and established a milestone in the modern microbiology and molecular microbiology. So the first one where we want to talk is in 1928, F. Griffiths. His very important research was to see Streptococcus pneumoniae in mice. So what happened is for Streptococcus pneumoniae, we know about that already. We learned in the exam too. We talk about lots of them. There are different types. One type, the capsule is smooth. Another type of the chemical, the capsule is relatively, we call it rough. Now the smooth one is a pathogen. What I mean the pathogen, it will cause pneumonia and the mice will be dead. The rough one, we call it a surrogate, which means the mice can survive. So he did a lot of the study, and there's a serious important study, if you remember. So I first inoculated mice with a smooth pathogen. So mice is dead. And then I inoculated the mice with a rough one. Okay, what happened is the mice survived. And then what we do is we do a heated smooth sample. Okay, this heated smooth Streptococcus pneumoniae go to the mice. What happened? The mice survived because the pathogenic chemical has been injured. And what he did, the last one is very important, is a smooth one and a rough one. And the smooth is also heat injured, which is he did a smooth sample with a rough one, and then inoculated a mice. And what happened is the mice is dead. So the last example tells us there is some transferring here. Following this transformation principle, there is some chemicals transfer the genetic information and the pathogenicity from the heated smooth Streptococcus pneumoniae and go to the rough one, and it makes it pathogenic. But by the time, he doesn't know what is that. Now we know they do a guess. That could be DNA, that could be RNA, that also could be protein, or some other macromolecules. They can just guess. And the F. Griffiths study, Frederick Griffiths study, is basically focused on the animals. So thousands of the mice has been used and has been killed during his research and studies. But unfortunately, he hadn't finished his work because he was killed by the German bombing during the Second World War in 1941. And he was killed just working in the lab. So his work has not been finished, but people continue to do. So let's talk about next one is in 1943. Okay, everybody should know this time around is Second World War, and everybody is busy. But there is a 3 American scientists. Avery, McCarthy, and McLean in the Rockefeller Institution of Medical Science, which is right now is part of the affiliation with New York University of New York University. It's by the time it's called a Rockefeller Institution of Biomedical Research. They are trying to figure out what are the chemicals has transformation. And they do the research without animals and on the tubes. So that is a big breakthrough, because instead of using animals, you have to do what we call it in vivo research. This is already in vitro. So which saves lots of the energy and the budgets. So what they do is that they also combine with rough one and the smooth one. And this is also the heat engine. And by the time they are combining using different enzymes, which they're using RNase, DNase, and also protease. With these different combinations, they find that the only time the transformation happens, the only time the transformation did not happen is using the DNase, which means the DNA has been enzymatically hydrolyzed. If you use RNase and protease, the transformation still happens. And of course, the results, they do the streak plating onto the other plates. And finally, they find that information is DNA. And they also get a pretty good, we say it's a purified DNA. And this group did a big contribution in the scientific area. Avery passed away in 1953, unfortunately. So the work did not continue, but some other scientists also, they did a great work. So we using the tube study, based on the enzyme reaction, and we find that the transformation principal chemical is DNA. We already know that. But you know the research, what we call research. So we want to say this again. What means research? That is search. Okay, then you read, which means you have to be doing repeated. Which means other people using different methods or the similar methods couldn't get to find the same results. And this is exactly happened during the early stage of the DNA. There is another very important and famous study did by Hershey-Chase. It's called Hershey-Chase studies. So Hershey-Chase experiment, we just say. This is not candy bar, okay. Not Hershey chocolate. This is not Chase Bank. This is Alfred Hershey and Martha Chase. I believe Alfred Hershey is still there, and he is later on the professor in the University of Oregon. Last time I do the search for that. This is a study did in like 1952 around. So they, from a different perspective, to verify the transformation principal chemical is DNA, what they're using is a bacteriophage. So bacteriophage looks like this. And they also using a T4 phage, which is a template with a temperate phage. Phage is a virus. You should know that. It's specific for bacteria, and they have two very important but simplified structure. The outside is protein, and the inside is DNA or RNA. Here they pick the one which is T4 phage, it is DNA. And we know that protein and DNA, they have some very special chemicals. So what are the very special chemicals for the protein? It's sulfide. And they are labeled them using so-called radiation isotope. So it's not a general sulfide, it's S35. And the DNA also have a very basic and important, unique chemical is phosphates. And we're going to talk about structure real quick, okay? So they also do the isotope radioactivity labeling is 32. And when they do that, they're going to start to do is let the bacteria invaded by a bacteriophage, a virus. Now, a virus invaded a bacteria, it could kill a bacteria, which is bursted. That is we use these days a lot for the biocontrol for the bacteria. We're going to talk later on. But with T4 phage, what happened is instead of you killing a bacteria, the DNA could be inserted into the bacteria chromosome. But of course, some of them will cause the bacteria to die. And finally, what happened? The virus will also could using the bacteria chemicals to biosynthesize their daughter phage. So it's kind of a mixing with everything. But for T4 phage, majority of them, more than 70%, is integrated into the bacteria chromosome. So how they do it? They have the bacteria, they have the virus. After they're doing the research, they put them together. And they do the centrifuge. Now by the time, they do not have a centrifuge machine. Not like right now, we have a high-speed centrifuge machine can do like a high-speed centrifuge, spin down very quickly. They using a very general kitchen blender to do a blender just. And we know that most of the time, the virus here is phage, is on the top. And the bacteria, they are at the bottom. And so they get the precipitation from the KitchenAid blended solution. And in the bacteria, they find it existing is P32, not S35. So what this tells you? This tells you DNA is possibly integrated into the bacterial genome, and they carry on the transformation genetic information, which means it's followed transformation principle. But protein doesn't have the same function. Okay, so that's from the phage and the bacteria. We say it's from microbiology standpoint, verify that the DNA is an important chemical which has transformation principle. So then the time goes. That we're going to talk about is 1953. Okay, people get to know the structure of the DNA. We first know the chemical, but then we need to know the structure. This is an important and the milestone, and the major milestone in the microbiology or biology history is two great scientists, Francisco Crick and James Watson. Crick is still there. James Watson, let me just double check. Sorry, James Watson is still there. He still lived, still survived. And Francisco Crick passed away, very long life. So first of all, James Watson, he is a newly graduated PhD student from University of Indiana. And when he got a PhD, he tried to do a study abroad. So he go to England, go to the Cambridge University in the Cavendish Research Lab, and find Crick. Francisco Crick, by the time, is working as a physician. It's a doctor in the Royal Community for the British Britain. So they get along with each other. Although they have like 12-year-old, the age difference. It's a decades old. But they get along with each other, they talk every day. I remember, if you look at history, James Watson liked birds. So they talk about birds all the time, but also they talk about biochemistry. By the time, with the limited available of the research facilities, those we call it's a theoretical biochemists. It's no longer have those type of discipline, because right now biochemistry is a heavily experimental based discipline. No longer you can sit and talk, have a cup of tea, you're going to create something. But the time is different. Okay, they met up each other, and it's very luckily they talk every day. And one day, there is an important lady, Rosalind Franklin. Rosalind Franklin is a research associate in Morris Wilkins lab. Rosalind Franklin did important research. What she did? She showed them using X-ray diffraction photographer find that we already know it is possible double helix. It's very lucky. But by the time they already know it's double helix, they already know the DNA have ATGC. A compromised with T, G compromised with C. They already know, they're already getting there. But the problem is that why they cannot make a final determination, the reason is they don't know the ratio between them. Because if the ratio doesn't work, this double helix structure may not be established. By the time they met another great scientist, Erwin Shograf. This is the chemists in Columbia University. Shograf shared his most recent finding, we know, called Shograf rule. What are the Shograf rules? The ratio of A to T and G to C is always 1:1. So in other words, in the different species, you may have a different amount of the A, different amount of the A and T, and different amount of the G and C, but their ratio of A and T and G and C is always 1:1. Then bingo, this kind of works. So in 1953, they published a single most beautiful paper in the modern history of molecular microbiology. It's only 1.5 pages. Where it is, I did it for you, right here. You can download and you can read it. It's only 1 page. Molecular structure of nucleic acids. Double helix structures it. That's a page. And in the second page, this is what happened. Basically 1 page. 1.5 page with a picture. This is Rosalind Franklin shared them the X-ray diffraction, where they find the double helix. It's only 1 page. And the people say published in April 25th, 1953. And we all say this is the single most beautiful paper in the 21st century, which they established the baseline, a big milestone in the molecular microbiology area. And after they have this publication, and they go to celebration, and they go to the pub. That is a very traditional way they celebrate. They go to the pub. They buy everybody a beer. And in the pub, they told everybody what they found. Their discovery, although nobody is understanding. So today, if you go to Cambridge University, suburb of London, they have one pub called the Eagle Pub. In the Eagle Pub, they will have a plate which is saying James and Watson have the DNA discovery right here. One of the students, a couple of years ago, showed me the picture. She had a study abroad with a health science center with WVU, and they went there and have a trip. So it's really true. It's very amazing for the breakthrough. Okay, so that is what the story about for the discovery of the DNA. So now we talk about the history already. So then we're going to go ahead to talk about the DNA structure. And you may know, if you know, we just do the review, but something we want to emphasize if you're not very clear. So we want to go, this paper you can read. It's a brief paper, but it is important. So we want to talk about what's the DNA structure, what it looks like. So this section we're going to tear it off. So first of all, what is the DNA called? What is called? Deoxynucleic acids, that's called DNA. What's the structure? First of all, in the center is a ribose. But it's a special ribose, what we call. It's a deoxyribose. So what means deoxyribose? Carbon number 1, carbon number 2, number 3, number 4, number 5. At carbon number 2, instead of I have OH, this is gone. That's called deoxyribose. So that's the center. At the carbon number 1, we add a chemical. This is what we call base. And at the carbon number 5, what we do, we already talked. Everybody knows it. A phosphate. So what's the major structure of a DNA? A ribose, deoxyribose, a phosphate, and a base. So I always say DNA is a very boring chemical. Because only 3 things, a base, a deoxyribose, and a phosphate. But make the people working every day, so a boring chemical make you prolific life. So compared to the protein, this is very boring. Protein, you have amino acids, you have a peptides, you have a primary, secondary, tertiary, quaternary structure. This is only that. Now what are the base? Base we have A, T, G, and C. We already know what they call them. So if they all have been composed with a 6-carbon ring with a 5-carbon ring, this is we call purine. What are the examples? Yes, adenine and guanine. And sometimes we only have one 6-carbon. This is what we call the pyrimidine. And it is composed by T and C, which is thymine and cytosine. And we also know at RNA, it's very special. Instead of we have thymine, we have uracil. And this is you already know, but do you know the structure? Let's talk about that. So what is the A looks like? So we want to go a little bit deeper. Okay, that you already learned in biology class a long time ago. We don't need to be talk these things early. What is A looks like? So we already talked about it. 6-carbon, 5-carbon. Okay, not that one. NH2. That's called adenine. Okay, what is guanine? Since this is all purine, so guanine structure and adenine are basically all the same. Very similar. Very, very similar. You don't even have to worry about this side. This is the same thing. The only difference is here. That's all. So that's guanine. Okay, what is thymine? That's a little easy, 6-carbon. So what does thymine looks like? Right here. That's thymine. So what does cytosine looks like? Little similar. Not a big difference. So we have so that's thymine. That's cytosine. Now, what's a uracil? How we label here? 1, 2, 3, 4, 5, 6, 7, 8, 9. So remember, ATP connected with a 9 units of the adenine. How about here? 1, 2, 3, 4, 5, 6. Thymine and the uracil are basically the same. The only difference is CH3 replaced with H. So we call H, which the proton replaced with a methyl group at an unimportant place, which is a position number 5. That is not important. So this is important for this structure. Okay, how they connected with each other? A complement with T, G complement with C. What is this called? Hydrogen bonds. Hydrogen bonds is relatively weak bond, so they can break down. Now, by the way, the GC, G complementary with C, they have 3 hydrogen bonds. So, when we design the DNA primer for the PCR reaction, we need to thinking about components, a concentration of the GC because they have more hydrogen bonds. What are the other 2 chemical bonds you learned in your chemistry class? Except besides the hydrogen bonds. Ionic bonds you learn, is that right? So you have, that's ionic bonds. Covalent bonds, did you learn in your class, chemistry class? That's covalent bonds. That's angle. That's covalent bonds. And the plus hydrogen bonds, 3 most important bonds. Okay, so that's all the basic knowledge. So what is the double helix structure? Like this. And they run a different direction. So 5 prime, 2, 3 prime, 3 prime, 2, 5 prime, and they are composed with A and T and G and C. If we want to talk a little bit of structure, what's the distance here? It's 2 nanometer. Okay, what is the 1 ton here? That is around 10 BP base pair, 10.5 BP base pair. This, how many? 0.34 nanometer, okay? And we call this is a minor growth, this is a major growth, and all these things we can talk about. So that's the DNA structure. It's a double helix. We call it double helix and anti-parallel. Okay, so that's the DNA structure, what it looks like, and the DNA, the base, also the chemicals we talked about already. Here is the slides, what you can see, what they gave you the example, okay, when they talk about is, if you have a base and the pure and the ribose, they combine, they call it a nucleotide, side. So with a phosphate, we call it a nucleotide, and when they're going through the polymerization, we call it nucleic acids. Okay, that's pretty straightforward. Now, let's talk about the thing, how the replication happens. Anti-parallel, that's, you already see it. These already talked, okay, DNA replication. DNA replication, it is called semi-conservative. Which means they first of all they separated using the mother bands as a template based on gene complementary policy to have their daughter bands synthesized. In the history, when they talk about the semi-conservative methods, there is another research to prove and verify it. It's called Matt Mendelson, so we call it a Mendelson Starr research. So that's Matt Mendelson and Frank Starr, are 2 brilliant graduate students in the Caltech University back in 1950s. So Caltech, you know about that. If you see the Big Bang Theory, all those geniuses coming from that university. It's a private university in the Central Valley of California. So Matt Mendelson and Frank Starr research, what they do is you have a DNA double helix. I'm going to label them using isotope, but using original nitrite, which is N14. And then what happened is, I'm growing, let the bacteria grow on the agar media. This agar media is filled up with N15 instead of N14. So what happened? And we know they're going to be separated. And N15 going to go there. And then later on what happened? And you know that this going to go, this will be separated, this will be separated, and this will goes. And then what happened? Semi-conservative, semi-conservative. This is N14, this is N14, but in the middle, these are all N15. So with more and more replication, and we know the bacteria is generation time is doubling. It's very quickly can do. More and more will be N15 compared to N14. So what they do, they're using a tube, and this tube they are adding Sicilian chloride. So using Sicilian chloride could cause gradient centrifuge, and they will see a different line. And what they find, more than 90% it is N15 at the bottom, and on the top, roughly only 10%, which is N14. And with more generation time, this N14 amount will be decreased dramatically. So this is verified, the semi-conservative replication of DNA. Okay, so that's another very, very important research, how they started to work. So then we want to spend in time, talk about the DNA replication, how that works. And especially the enzyme activity, how that goes. Okay, so we're going to move these. We left one space, and we want to talk about it. By the way, there is one thing I forgot to mention. So 1953, James Watson and Francisco Crick, they published this paper. And in 1963, they both awarded the Nobel Prize to award their credibility. At the same time, Maurice Wilkins also awarded the Nobel Prize with them. Because Maurice Wilkins is the boss of Rosalind Franklin. The Nobel Prize cannot award it to the person who already passed away, generally speaking. Rosalind Franklin, unfortunately, in 1958, she was passed away because of the ovoid cancer. And once they published the paper, when they get the results, and Crick and Watson, they celebrated with Rosalind Franklin. They took her to have a trip around the world. But unfortunately, Rosalind Franklin passed away much earlier, so she didn't get she didn't get the award, but she did get a recognition in the history, which is she did that important X-ray diffraction to find the double helix. So there is a little bit of story behind that. We're going to talk about the next one is the DNA replication. So DNA replication, how are we going to do? For bacteria, so we are going to talk about the bacteria. The DNA replication happened in the place called replication fork. So Originally, they are twisted, like that. So that's why I didn't remove that, like that, highly twisted. And in one point, there is a called a replication fork. They started to generate. We also call it OIC. Then they start to formation, and they start to do the replication. At the beginning, they need a couple of the enzymes, which is we call it a DNA group. So we have DNA A, then a B, and then a C. So what is a DNA enzyme for? This is we call it an initiator. So initiate the DNA replication. What is the DNA B enzyme? This is a helicase. So when you look at the words, what means a helicase? This is a double helicase, anti-parallel. I have to break it. So I need an enzyme called helicase. Now what is the DNA C enzyme? This is the holder. So we say it's a helper, holder DNA B, which is totally on helicase. So once the very twisted a double helicase has been formed, and they break down because of the weaker hydrogen bonds, but from the physics standpoint, it's very easy to twist it back. So we need something to let them stay separated. Therefore, we had those chemical compounds attached to them. This is called SSB. Single strand binding protein. So they started to do. Okay, that's a preparation. Now, DNA have two different strands. 3' to 5', and 5' to 3'. DNA replication. You should always know they need an enzyme, obviously called DNA polymerase, because the replication is a polymerization process. DNA polymerase have A, 1, 2, 3, 4, and 5, based on the different molecular weights. But the more important is DNA polymerase 1 and 3, and the most important is DNA polymerase 3. The polymerization process is very quick. They can do 2,000 base pair per second. And who is the person discovered DNA polymerase is Arthur Cronenberg. This is a great American molecular microbiologist, and he also awarded a Nobel Prize in the 1970s. His son, Roger Cronenberg, is also very good, and he discovered the RNA polymerase. It was a Nobel Prize in 1994. So the family is very intelligent, to be honest, so that's very good. So DNA polymerase. So you have 3' to 5', and 5' to 3'. You always remember, the polymerization direction is 5' to 3'. Now, what this means, this is lots of people is confusing about that. What means 5' to 3'? This means the newly synthesized band is synthesized from 5' to 3'. So this guy to 3'. Go that direction. That's what it means. In other words, it's reading the mother bands from 3' to 5'. Okay, that one means the direction from 5' to 3'. So let's give an example. 3' to 5'. The top band, let's say I have A, T, G, C, T, A, G, C, let's say. Now, usually, if you don't understand or you're not very good at genetics, 3', I usually add OH there. And the 5', I add a P there. So I usually wrote like this. Okay, based on gene complementary policy, you should know what the new band synthesizes. That is T, A, C, G, A, T, C, G. Okay, what are they? This is P, this is P, this is P, P. At this end, we're going to add a OH. So this could be long and long, this is long and long, and then in the cell membrane system, you always have like A. They carry on 3-phosphate. They come in based on gene complementary policy. Of course, they need energy. They hydrolyze, use energy. Sometimes GTP, sometimes TTP. Now, at the very beginning, when they started to do the synthesis, they also need help. They need a very small segments of primers. That is RNA primer. And the primer is composed by primaries. So it is very interesting and very ironic. So also Cronenberg awarded the Nobel Prize for founding discovery DNA polymerase. But later on, about 20 years later, people find this is not really a DNA polymerase. This is actually the RNA primerase at the beginning to synthesize RNA primer. But the principles are the same, so they still give much credit for him to have that discovery. But honestly, that's not a typical DNA polymerase. It is more like is an RNA primerase at the beginning. So this can happen naturally. We call leading band. How about here? We get the problem. So I'll show you what's the problem. Remember, the replication is always 5' to 3'. What this means, that is why I always say we have to know the chemistry structure. That is the reason. Okay, we have this. This is base. This is H. This is H. This is OH. This is H. This is CH2. This is O, phosphate. Phosphates. Okay, when you go here, what's the second one goes? Goes right here. Then where they go? Okay, where they go? That's a base. By the way, this is H. So H, 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 OH. 5'. So 1', 2', 3', 4', and 5'. 3'. They go this way. That is 5' to 3'. What this tells you, every time you must have this free available hydroxy bonds showing up, or hydroxy chemical showing up. Go this direction, there's nothing you can do, because there's no free available hydroxy bonds. So what they do, remember, the newly synthesized, you can only do that way. So they have to synthesize some very small segments. And these small segments is actually very similar to here. It is composed by the RNA, so RNA primer. And they have to do this. And who is the person find it? That's Okazaki, so-called Okazaki segment. Okazaki, yeah, that's kind of, it's very short life. People say if Okazaki has been lived along another 10 years, he could be awarded the Nobel Prize. Only 44 years old. So what we do, these primer has to cut. This rely on DNA polymerase 1. Now once they cut, what happens? There is a niche there. Is that right? There are lots of the niche comes. So you have to seal it. You have to seal these. Seal this niche, you need using the DNA ligase to connect it. Then more and more you synthesized and there's a replication finished. Okay, once the replication finished, we get some problem. So not done yet. We finished, but they have something we have to solve, the problem. And remember, bacteria system is very magic. Think about that. In the very short lifetime, they can do so many work, so much work. So once you synthesize what you have, there's a two issues. Number one, top logical issue. So think about it. They twisted it. They separated. So they twist it again. So the tension is higher. So what's it? It become like that. Could be. Like this. This is not, from the energy standpoint, this is not available to be used. Or they can continue their life because the tension is too high. So we call it positive tension. Or we say positive supercoil. Now the positive supercoil needs to be released, become a relaxed double helix. So that is we call negative supercoil. That's the first issue to have to solve. But very lucky, the bacteria have the enzyme for that, which is called topoisomerase. Can relieve the positive supercoil to become negative supercoil. Now, topoisomerase, they also have 1, 2, 3, based on the different molecular structure and the weight. Typically in E. coli, it is called DNA gyrase. So the first issue solved. We have topoisomerase. The second issue we call fidelity issue. So think about it. Twisted, opened, twisted again, release the tension. We cannot make sure every single gene base complementary with each other is correct. Same as you write an essay, like write a 2,000 words or 10 pages essay, you cannot make sure all your spelling is correct. So that's a fidelity issue. And it is huge. So first of all, the natural error happened for a bacteria is usually 2 errors per doubling time. And the people did the calculation. This is approximately 10 to the power of minus 3% mistake. That is very high. Genetically, we cannot allow these to happen. This is 1 among 1,000 the mistakes happen. That's very high. So, bacteria can reduce the mistake. We had DNA polymerase 3. DNA polymerase 3 can do a proofreading. So they will be removed through a scanning to remove mismatch base pair. So for example, okay, we have ATGC and we have TACG. Let's say we have TAGG. They could cut it and then add the correct one C, and these can be sealed with DNA ligase. And the DNA polymerase 3's proofreading process will reduce the mistake rate to 10 to the power of minus 5 to 10 to the power of minus 6, about 1 per 100,000 to 1 million cells. Is this solve the problem? Not really. It's still too high. So lots of our cancer caused by the mutagens or mutations. Part of the reason is that, let's say the colon cancer. It's part of the major reason is the DNA mutation happens. So we need another thing to solve the problem is called mismatch repair system. The mismatch repair system, lots of the time what we do, in addition to that, we, let's say ATGCCGA, and we have TG, let's say TG, CGGCT. What we do, we label them with a methyl group. And we cut them, then we synthesize with A. So using a methyl group mismatch system could reduce the error to this. That's very minimal. Okay. Now, in the certain circumstances, if the bacteria cells, the mutation is too high, they have to initiate SOS recovery system. That has to be activated. DNA polymerase 4 and 5. So, this is all I have today, basically, for DNA structure and the DNA replication. What are the key things you need to know? Is right here, this slide. That is important for the procedure in your mind, what they're going to do. And more important is this slide. The enzyme function, you at least on your mind, you need to briefly know what's going on. And this is well explained to you. What happened for the lagging bands, we need Okazaki segments. Can you see it? If we don't have a free available hydroxy bonds, like here, it will not happen. Okay, so that's all I have.